Hi, my name is Ahmad, and I'm going to talk about growth of salmonella in no-knead bread dough. Introduction. A convenient bread making method involved prolonged fermentation of no-knead dough has become popular in recent years. This process, a mixture of yeast, salt, flour, and water, are allowed to stand for 12 to 24 hours at room temperature. This is the process to make the dough, at which time additional flour is added, a loaf is shaped, and the rising and baking process continue. The extended rise time presents certain food safety challenges. Foodborne pathogens such as Salmonella enterica can grow in fermenting dough used in making teff injera, a pancake-like fermented bread. And here we have a picture of teff injera, a pancake-like. Consumers have been advised to avoid sharing the so-called friendship bread, which is the second type. It has been exposed to in controlled temperature for an extended period. The mixture is allowed to stand at room temperature for up to 10 days. The prolonged fermentation at room temperature is necessary for the production of the characteristic tangy quality of the starter, but it raises food safety concerns due to the presence of milk and the heightened concern over the safety of bakery dough in general. Also, in order to control and prevent pathogen proliferation that leads to serious kitchen cross-contamination or disease outbreak, food safety rules and regulation in US do not allow food handlers to leave food items that vulnerable to the growth of foodborne pathogens in the temperature zone 5 to 57 degree for more than 5 hours. Now we're going to talk about the method. On our study, or in this study, a study was challenged the safety of no-knead dough fermentation practice in the presence of artificial inoculated foodborne pathogen Salmonella enterica as an infectious agent. So it has been used on this study, Salmonella enterica, in order to examine the effect of fermentation temperature and the growth of the pathogens in no-knead dough. So here we have inoculum preparation for serotype of Salmonella were grown and then subsequently centrifuged and pooled in sterile tap water. And this is the picture of Salmonella grown on XLD agar. The inoculated water was used for bread dough preparation. Now, bread dough preparation. Bread dough mixture were made with inoculated or non-inoculated water control, which is controlled. Non-inoculated water is the control one. The mixture was formulated according to a highly publicized recipe. The ingredients were placed in a sterile bag and mixed bag by massaging the bag. 3 brands of yeast were used independently for ferment no-knead dough at 21 Celsius for 6, 12, 18, or 24 hours. The 3rd yeast was used to evaluate the effect of fermentation temperature on pathogen growth at different temperature. Separate bag of dough mixture was prepared for each sampling and temperature. Then we have a microbial testing. After fermentation, each dough mixture was diluted with equal amount of water and homogenized in a laboratory blender for 1 minute. After that, seal dilution prepared and spread plate on agar. We did spread plating on agar. After that, this agar put in incubator for 24 for 48 hours at 36 Celsius. Salmonella colonies were counted and confirmed as Salmonella with biochemical test system. Then statistical significant were analyzed. Now the results. Microbial growth in no-knead dough. Significant growth of Salmonella was found in dough fermented for 12 hours by 2 type of yeast or 18 hours by other type of yeast. Salmonella enterica multiplied continuously in the no-knead dough system and resulted in a 2 to 2.6 log increase over 24 hours period. Temperature effect, no pathogen growth. Significant growth of Salmonella occurred after dough fermentation for 6 hours at 27 to 38 Celsius or after 12 hours at 12 Celsius. This is growth of Salmonella. Also, Salmonella growth in the initial 12 hours was positively correlated with fermentation temperature. Okay. Salmonella count increased by 3 log unit after 12 and 18 hours at 38 and 32 Celsius degree respectively. Salmonella count in no-knead dough was obtained after 18 hours of incubation at 32 Celsius. Now the discussion. Foodborne pathogens such as Salmonella enterica is associated with raw food material, commonly used in domestic kitchen. Also, both healthy and ill individuals can introduce this human pathogen to food when proper hygienic practices are not used. Okay. The researcher found that low levels of introduced Salmonella enterica can multiply significantly overnight in no-knead slow rise dough despite the presence of high levels of active uh, fermenting yeast. The food safety concern associated with the possible growth of Salmonella in no-knead dough is similar to that of thawing raw poultry at room temperature. Also, bread is vulnerable to post-baking contamination and has been associated with outbreak of Salmonella infection. According to the US FDA, as few as 15 to 20 Salmonella cells can cause salmonellosis. This low infectious dose and the observed growth potential of Salmonella enterica to 5 log CFU per gram during fermentation in no-knead dough suggests that recipes exposing raw dough to room temperature overnight may pose a food safety risk to consumer. Future research to develop and validate no-knead bread recipes that do not promote the growth of food-borne pathogens is recommended. And this is my reference. Thank you, and if you have any question, please let me know. Bye bye.